everyone, and welcome to our 10x-pert workshop for tumor dissociation for single-cell RNA-seq. I'm Shauna, the 10x Community Manager and your moderator for today. Before we get started, let's go over some quick housekeeping items. Our 10x-perts who I'll introduce in a minute will be giving a presentation followed by an open Q&A session. We're recording this session and we'll make the recording available on the 10x community shortly. Please note that all attendees are on mute, and if you'd like to ask a question, please select the Q&A option on the bottom of your screen. Selecting the Q&A option will open a box where you can type your question. Please note that you can ask your question anonymously by checking the Send Anonymously option. And finally, if you're interested in tweeting during the session, please use the hashtag 10xpert. Okay, now let's meet your 10xpert. Our host today is Jill Hirschleb, the sample prep group leader here at 10x. Jill has a PhD in cellular and molecular biology and has spent the last 15 years working on technology development for high-resolution genome analysis, including optical mapping, long-read sequencing, and precision molecular counting. A fun fact about Jill is that she's been working with high molecular weight DNA since 2003. Joining Jill will be Sharmila Chatterjee. Sharmila is a scientist at 10x and is currently a member of the sample prep group, where she is dedicated to developing new demonstrative protocols for the chromium single-cell gene expression solution. She holds a PhD in physiology and has worked on different aspects of cell biology, including using the mouse as a model system to study developmental biology. A fun fact about Sharmila is that she developed our demonstrative protocol for nuclei isolation. And now I'm going to hand it over to Jill so she can get started with the presentation.
Hi, everyone, and thanks for joining on the call today. Today, we're going to be focused on tumor dissociation with use with our single-cell gene expression solution. So if you are tuning in today, I'm going to assume you have some familiarity with our workflow for single-cell gene expression. If not, if you're new to single-cell genomics, I encourage you to visit our website where we have a bunch of great introductory and training materials. So I'm going to skip over the nuts and bolts of how the 10x product works. But I will say that in order to do single-cell gene expression, you need to start with a single-cell suspension or single nuclei suspension. And I know this may seem obvious to a lot of people, but it's worth restating. And it's not just any single-cell suspension. You really need a high-quality cell suspension. Sample quality is absolutely critical for generating best quality single-cell genomic data. So the cell suspension needs to be clean, meaning the cells are well singulated and free of clumps and debris. When you're working with intact cells, you need high viability. The cells need to be very viable. And the main reason for this is that dead and dying cells can release their RNA content into the solution, and you won't get back a useful or rich transcriptomic data set from a dead or dying cell. And finally, the cells have to be handled gently, which includes pipetting them slowly, keeping the cells on ice, and working quickly. And there are challenges to achieving these three attributes of a high-quality cell suspension. It's difficult to dissociate tissue uniformly. You can oftentimes get aggregates or clumps forming, and you can have free-floating mRNA, again, and these will all lead to a reduction in data quality. Some challenges when working with less than viable cells is that, again, the dying cells have reduced RNA content, and you want to minimize that as much as possible. When it comes to cell handling, you need to work on your pipetting technique. You need to choose the appropriate buffer to keep your cells happy. Even though there are a bunch of challenges towards high-quality sample prep, that's okay, because at 10x we've developed solutions to each of these challenges. And they really require the right methods, good products, and then honestly, a little bit of practice if you're not used to doing this type of work. So on our website, support.10xgenomics.com, we are dedicated to helping you all out by providing different types of advice and protocols for doing single-cell gene expression. So all of our protocols are free to download, and if you're very new to this, we suggest downloading our flowchart, which can walk you through single-cell sample prep. Some of the resources we have available include general guidelines for sample prep, And I'll draw your attention here to the first one, tumor dissociation for single-cell RNA sequencing. That's going to be the main topic of today's presentation. In addition to general sample prep guidelines, we also have very specific sample types, dissociation of embryonic mouse neural tissue, fresh frozen human PBMCs, and we also have sample improvement protocols, including enrichment of certain cell types or depletion of dead cells from a single cell suspension. Throughout the course of my group's work, again, we're really focused on sample prep solutions. We ended up buying a lot of products from Miltenyi, so much so that we thought it was natural that our company should work together. Again, we're both focused on providing really high quality products for cellular analysis. So the agenda for today's presentation, we will be going over tumor dissociation, both a study design and a workflow. And then I'll go over how to look for hallmarks of sample quality in your single cell genomic data sets using both Cell Ranger and Loop Cell Browser. And those are software packages available from 10x. And we'll follow this up with a lot of time left over for Q&A. So on the phone today, we also have Cole Jones and Murray Francis from Miltenyi who will be joining us for the Q&A. And when it comes to this tumor dissociation workflow, we all work together to generate these data sets. And from the 10x side, Sharmila Chatterjee and Elena Puleo did most of the work in the study, so I'd like to acknowledge them today. So for the study design, we decided to work with syngeneic mouse tumor models. And for those of you that aren't familiar with this tumor model, how this tumor model is generated is that you have a mouse which develops a tumor, you excise the tumor, and you transform that into cell lines in vitro. So you culture up a tumor cell line, which you can then re-implant into different mice of the same inbred strain. And in this way, you can generate a reproducible model of disease. And since one of our study goals was to look at reproducibility, this is a nice system for doing so. And we took 3 different well-known syngeneic mouse tumor models from breast, colon, as well as melanoma. From each of these 3 cancer models, we obtained tumors from 3 independent mice. So that's a total of 9 tumors. And we processed all of these with our single cell gene expression solution. And we processed them at different steps along the sample prep workflow, which I'll explain a little bit on the next slide. So all in all, we sequenced 9 tumors from 3 different tissue types, 3 replicates of each, so a total of 9 tumors. We generated 47 single cell sequencing libraries, and we sequenced more than 84,000 cells over the course of this study. And how the workflow, the way the workflow, the description of the workflow follows. So first is dissociation, and to achieve dissociation, we use products from Miltenyi. So the octo dissociator with heater is an instrument from Miltenyi, which enables enzymatic and mechanical tissue dissociation. And shown here is a picture on the bottom. How it works is that you have these plastic tubes which contain a turning, almost a pestle. And what you do is you add your tumor piece to the tube, and you add an enzyme cocktail, and we use the mouse tumor dissociation kit from Miltenyi for this purpose. You generate a nice homogenate. You plug the tube into the instrument and you run a preset program, and you end up with this almost slurry. So it's not quite a single cell suspension at this stage. The next thing you do is filter the cell suspension, and this is important for 2 reasons. One, you want to get rid of any large pieces of debris, which you can see here. Sometimes there are large chunks that are left behind, but you also want to get rid of small clusters, clumps, and doublets, because it's really important to generate a truly single cell suspension. And this is one of the stages where sample preparation for single cell genomics and sample preparation for flow cytometry can kind of diverge. So with flow cytometry, you can gate out doublets and gate out a little bit of debris. So you can sort of run a selection when you're on the instrument. With single cell genomics, with our solution, everything in your cell suspension is encapsulated, which is why it's very, very important to spend the work upstream working on your sample prep. We tested out 2 additional sample cleanup steps in the course of this study. One is a red blood cell lysis solution from Miltenyi, and the next is a dead cell removal kit from Miltenyi. And the way the dead cell removal kit works is that you get a vial of magnetic beads, which have epitopes that recognize dead or dying cells. You pass your cell suspension over a magnetized column. The dead cells remain bound to the column, and the effluent contains your live cell fraction. So we wanted to, our goals for the study were really to execute on this workflow multiple times, to take the sample through various stages of cleanup, dissociate, filter, sequence it, lyse the red blood cells, sequence again, clean up the dead cells, sequence again. And so our goals were to look at absolute performance of our single cell gene expression solution. We wanted to look at reproducibility of these dissociated tumor samples, and we wanted to look at the ability to characterize the heterogeneity of each sample. And the way we look for each of these attributes of performance, reproducibility, and heterogeneity, we turn to our 10x software products. So when you run our software package called Cell Ranger, you get back a number of different metrics that inform about your experiment. You'll get metrics that can tell you how the sequencing run went, attributes of what happened on the Illumina flow cell. You'll get metrics that tell you about the sequencing library itself, what's the insert size, the duplication rate, and you'll also get metrics back that report on the quality of the starting sample. And I'll focus on a few of those today to help. And the purpose of walking through some of this is to help all of you guys interpret your own data sets when you get them back. So the first thing we look at is library cleanliness. And this metric refers to when you get a read back from your short read sequencer, does that read, can you confidently map it back and assign it to a single cell? Or was that read representative of a free-floating piece of RNA that was present in your cell suspension? And it's simply a ratio, the fraction of reads that you can map back to single cells. And you want that value to be as high as possible. Now, the way I've plotted the data here, I'm combining all 9 tumors. So I'm combining all 3 different tumor models, the breast, the colon, the melanoma, all of the replicates. And what I'm separating out on the x-axis is the different stages in the workflow, and this is in order. So stage A is the most preliminary stage of the sample prep workflow, immediately after dissociation and filtration. Column B is after red blood cell lysis, so adding one more cleanup step. And columns C and D are after dead cell removal, 1 or 2 rounds of dead cell removal. There were some samples that required 2 rounds to boost the viability up. And what you can see here is that over the course of the additional cleanups, you can see an increase in the library cleanliness metric. Immediately after dissociation, we have a median value of 78% of reads mapping back to single cells. And after all the cleanup, we have an average of 88% of the reads mapping to single cells. And in our opinion, all of these values are quite good. We have seen there's one outlier that came back with less than 50% of the reads mapping to cells. And you can oftentimes, it's probably sample dependent and probably protocol dependent, but there are some tissue sources which will give you a very low value here. We aim to be above 75% when we're working with primary samples. So all of these values were quite good, but this is just a demonstration that applying extra cleanups, you can boost that metric a little higher. The next thing we look at is library complexity. And this is the median number of genes detected per cell. And again, you want this value to be high. And it's the genes you detect per cell and the level of expression of each gene. Those are the parameters that you use to make your cell clusters and to sort of determine the identity of each cell. So it's an important metric. Again, you can see over the course of the subsequent cleanup steps, that value is increasing. Again, 2,000 genes per cell is a really great starting place. So we were happy with all of these values internally. And I should note that this metric is calculated at 20,000 reads per cell, which is very low sequencing depth. And for you guys listening on the other line, if you want to look at library complexity and really compare samples apples to apples, you need to normalize to the same number of reads per cell. Obviously, if you sequence more, you'll recover more genes. So if you want to make determinations about which sample had a more complex library, you need to normalize that. So where we found a moderate increase in library cleanliness and library complexity, we found a substantial increase in cell recovery over the course of adding the additional cleanup steps. And this does not mean cell recovery from the starting tumor. We got millions and millions and millions of cells back from the starting tumors. This is a measure of when you're running the 10x assay, you target a specific number of cells that you want to recover. And in the case of the colon tumor, which is shown here, we targeted 5,000 cells recovered. So the graph here, this is a new way of presenting the data. I've broken it up a little bit differently. Here, these are only the 3 colon tumors, and each of these lines represents one individual tumor. So you're seeing the cleanup steps on that specific tumor as we go through sample prep workflow. And here you can see by adding in the extra cleanups, including dead cell removal, that value of recovered cells is getting a lot closer to your target. And that's probably due to many different factors, but some of those factors include an increase in cell viability as you go through the cleanup workflow. If you're loading dead or dying cells onto the instrument, you're not going to get back a really rich transcriptome. It's almost like it will disappear into your experiment. If you have a dirty cell suspension, if you have debris, it's really easy to accidentally count that as a cell. And then some just cells that are fragile or lysing or in the process of dying will also not have a high RNA content. So as you get rid of those populations, your cell counting will be much more accurate, your recovery will be much higher. And of course, this will vary by sample type and by protocol used. But you can see that this trend holds true for the colon samples, for the melanoma samples, and for the breast samples. And there's definitely a heterogeneity in response here. But overall, the trend is pretty clear that with additional cleanup steps, your cell recovery tends to get more accurate. So next, I'll show you how we look at heterogeneity within a sample using Loop Cell Browser. And here we're looking for reproducibility. This is really what we wanted to evaluate in this controlled study, with how reproducible are these cell populations. So let's just look at the breast tumor data to start with. Here I've chosen to group the data 2 different ways. First way is looking at early in the workflow. So this is immediately after dissociation and filtration, no additional cleanup steps. And over here on the right, I've grouped together the samples that had red blood cell lysis and dead cell removal. And what I wanted to see was, do we see a difference in the cell populations, the frequency of cell types, and the ability of the samples to overlay each other? You know, did we see an increase in those properties as we add the additional cleanup steps? So what you're looking at here is a plot from Loop Cell Browser, and we'll go into this plot in a little more detail. Each dot represents a single cell, and this is graph-based clustering, also called a TSNE plot, which if any of you guys are familiar with single-cell genomics, you've seen these plots a bunch. And basically what it's doing is grouping similar cells together, cells with a similar transcriptome together. And what we would expect if all of the different samples analyzed were reproducible, and the samples here are denoted with different colors. If there's great reproducibility amongst all these samples, again, we're looking at 3 independent tumors from 3 different animals. The fact that the samples, that all of the, that the colors are intermixed uniformly means that there's high reproducibility among these different samples that we evaluated. We found that to be true at the early stage of the workflow, and again, after the additional rounds of red blood cell lysis and dead cell removal, which was a great result and a little surprising. I went into this thinking maybe we would find that after dissociation, we would find poor reproducibility, but after cleanup, we would then achieve a state of reproducibility. But for the breast tumors, we found that before and after filtering, the data looked quite uniform. And if you separate the data out so that you can see each individual library that was sequenced, and we have some technical replicates in here as well, the cell clusters are quite uniformly represented, even some of these smaller clusters are conserved pretty well across the different tumor samples. And again, we use Loop Cell Browser to do this analysis. We find the same thing with the colon tumor samples, that both before and after, with and without the additional cleanups, that the samples are pretty reproducible. But when it comes to the melanoma sample, this was the tumor type that really benefited from the additional cleanup. So if you look at the samples before and after dead cell removal, you can see that the pattern after the dead cell removal and the cleanups look reasonably similar across all three tumor types, all three tumors and the technical replicates. Before the additional cleanups, you can see two of the samples overlay each other nicely, and one is clearly divergent from the others. And this particular tumor model grows incredibly quickly within the mouse, is incredibly necrotic, and had markedly low viability when we received the tumors. And the one that was the biggest and the most necrotic is the one on the bottom, which is the standout. So again, this just goes to show you, if you have really poor viability, you definitely want to do a cleanup. And so now we'll do a little bit of a deep dive on one of these data sets to show you the type of heterogeneity that you can uncover when using single cell gene expression. So this is just the breast tumor data set. This is 6,335 cells. And this was the data set that went through the additional cleanup and filtering. And again, this is a view from Loop Cell Browser, and these are categories that are assigned based on graph-based clustering. So you go in without a hypothesis. A general rule of thumb is that if you see small, independent, or sort of geographically independent cell clusters, they're more likely to be a distinct cell type. Whereas within a big cluster, if you can detect substructure, that may be cell state, a metabolic state, proliferation state, or maybe a step along a maturation pathway. These are not hard and fast rules. This is going to vary based on the system you're working with, but if you're new to this type of analysis, that's a good way to think about this from a very general standpoint. So what you can do once you get this data back is you can go in with your hypotheses and pull out different clusters that correspond to well-known marker genes. And this is something which is very commonly done. If you look at CD45, you can pop out your immune cell clusters very easily. And if you then go in and look at the clusters with known markers, CD3, CD8, CD19, you can really easily assign the identity of your immune cell clusters. And then you can go explore different populations within the sample. So we have a small cluster here that could potentially represent fibroblasts, cancer-associated fibroblasts. They share well-known markers of cancer-associated fibroblast gene expression. If you dig a little deeper, we found a very small population that might correspond to mammary basal cells. But the dominant cluster here are the mammary tumor cells. If you're familiar with mammary biology, and we go back and forth here a little bit, the basal cells and the luminal cells, in a normal primary mouse mammary tissue, you'd expect to see large populations of both basal and luminal cells. However, this is not a normal primary mouse mammary tissue sample. This is a clonal tumor originating from a clonal cell line, which is a luminal tumor cell line. So this is actually what you would expect. You would only expect to see luminal tumor cells in this particular mouse model of cancer. So we find EpCAM, CLDN4, the KRT18, which are known to be expressed in the cell line for this particular breast cancer model. So again, this confirms the biology we would expect to see in this very particular tumor type. If you're familiar with our other data set, which is a tumor microenvironment study, we have a data set available online, which is from a late stage human primary cancer, in which you find a much higher diversity of cell types, including post-endothelial cells, vascular cells, more stromal involvement. You wouldn't expect to see that to such a degree in a mouse syngeneic tumor model. It's a much different type of tumor. It's a little less complex. So what we found actually matches our expectations really well. And then within these structures, you can still pick out some other substructure. For instance, shown here are different cell proliferation markers. So you can see within the tumor cluster, cells that might be actively dividing. And the nice or interesting thing is you can also see portions of the immune cell and fibroblast clusters also lighting up. So you can do this analysis on all the different tumor types. And so here I'm showing you the breast, the colon, and the melanoma tumors, both with and without the additional cleanups. And you can see that in the breast and colon samples, you can really easily detect this heterogeneity, both with and without the additional cleanups. The melanoma samples, again, because the viability was so low after dissociation, we didn't really get a lot of useful data. That was a sample type where we really, really benefited from the additional cleanup steps. And you can actually see some shifting proportions of cell types represented along with the additional cleanups. One example here is in the breast. These clusters down here are the immune cells, and up here are the immune cells. So you can see with the additional cleanup steps, in this case, we're selecting for the more sturdy, robust tumor cells. So another thing to keep in mind as you're designing your sample prep workflow is to keep in mind what cell types you're really most interested in studying, and maybe tune or optimize your workflow around that, because every additional operation will have a good effect, and it might have a slightly negative effect as well. Every time you spin or wash or handle cells, there's going to be some loss in your cells, which is just known. It's a known fact. So based on really what you want to see, it'll help you guide what you might want to do next time based on the cells you're most interested in. And so to sum all of this up, we found really good performance both before and after the additional cleanup steps. The column on the left is just after filtration, and the column on the right is after the additional cleanup steps. So really the significant changes were in the estimated number of cells, or the number of cells that you get back after running the system. So that's the metric that changed the most significantly with the cleanup. So again, it's up to the user to pick a sample workflow that best suits your application goals. But it's nice to know that you can intervene at various steps of the workflow quite successfully in this study. And once you have a highly viable clean cell suspension, you can actually run this suspension on any of our 10x products right now. So we did this on just one of the samples, so the colon tumor cell suspension, divided up the cells and processed them with our linked read genome sequencing product. And this text might be a little small to see on a laptop screen, but after isolating the high molecular weight DNA and running our linked read genome product, we got an average DNA molecule length of 144 kb, which is really significant considering that we're starting from a solid tumor sample. Next, you can obviously run the single cell gene expression solution. That was the focus of our talk today. Under development right now, we're actually taking the same samples and processing them with our single cell immune profiling solution. So we can generate paired T-cell alpha-beta chain information, as well as immunoglobulin heavy light chain B-cell information on the immune infiltrating population within these tumors. And that's under development right now, and hopefully we'll definitely share that as soon as the data are ready. And then finally, these samples were compatible with our brand new single cell CNV solution, which is designed to look at the heterogeneity present in solid tumor samples. And you can see down here, these are 4 lines representing 4 individual cells, and the flat lines are the 2 of the immune or host cells, and then the lines with the jagged profile are the tumor cells. And really something we found that was quite interesting is that when you look at a normal primary tumor with our single cell CNV product, you see subclones, you see a bunch of different heterogeneity within the tumor. And for this colon tumor model, because it's a syngeneic tumor model, it was perfectly clonal. So we had a very complex copy number profile that was identical across all of the cancer cells. Again, underscoring what you would expect to see from this type of tumor. And so with that, I will wrap it up, and we've left a lot of time to take questions from the listening audience. So please send them in via the chat function. And myself, Sharmila, Murray Francis and Cole Jones will be happy to answer any questions you have.
All right, thank you, Jill, for a great presentation. Before we get started with our Q&A session, I just wanted to introduce our two special guests from Miltenyi that Jill mentioned, Cole Jones and Murray Francis. They're here to help answer your questions today. To submit a question, please remember to select the Q&A box on the bottom of the screen, and we'll do our best to get through as many questions as possible. So we're going to get started with our first question. I understand cell viability is important, but is there a way to dissociate induced single-cell RNA-seq from FFPE tissues?
Hi, this is Cole from Miltenyi, and I'd also like to say thank you again to 10x for having us join this webinar. And to address this question, so we do have an FFPE tissue dissociation kit. Currently, it's been validated for dissociation of FFP carcinoma tissue, and for subsequent either kind of ploidy analysis with flow cytometry or sorting on tumor cells versus stromal cells, and then doing a bulk, then you could do bulk next generation sequencing on those separated tumor or stromal cells. When it comes to single cell sequencing from the dissociated FFPE tissue, this is something we're currently working on validating a process for, but we currently don't have a validated protocol for that yet. That is coming.
Okay, great, thank you. And the next question. You mentioned that during processing, cells should be frozen. Does that mean that my use of RPMI at 37 degrees with the Miltenyi dissociation protocol could lead to low count and viability?
I can take the question. So we mentioned to keep the dissociated cells on ice, and it does not need to be frozen. Keeping it cold is what the purpose is. But dissociation is at 37 degrees, and so doing the Miltenyi protocol with their required solutions at 37 degrees is perfectly fine for the tissues. Once the tissues are in that slurry or somewhat single cell state, we recommend keeping them in cold because processing takes time. Most people are not really working with a single piece of tissue. They're working with multiple pieces, and just handling takes time. So to overcome any degradation at this point, we recommend keeping single cell suspensions on ice.
All right, great. The next question. By using the octave dissociator, usually what's the cell viability rate after dissociation before removing the dead cells?
Okay, I'll take that. This is Murray from Miltenyi. There's going to be a lot of variability depending on the starting tissue. Even within tumors, there's a lot of variability between different tumors, different tumor types, amount of necrotic tissue. I have some general numbers here from the mouse dissociated kits. Overall, the tumors we've looked at, we generally get between 1.3 million to 2.3 times 10 to the 8, 230 million cells per gram. The viability for that is between 16% and 89%. And that's, again, over all the different tumors, all sorts of different amounts of necrotic tissue, things like that. For specific, like one type of tumor, the 4T1 tumors, the numbers are much narrower. Typically, again, for the 4T1, we get between 6 million and 18 million cells per gram of tissue, with a viability from 60 to 90%. There's going to, but depending on handling the starting tissue, there is going to be some variability, but those will hopefully give you some general numbers to base your work around.
Great, great, thanks. Next question. What if a portion of the cells are to be cultured? Can the cleanup process affect their growth?
I can take a stab at that as well. It's going to depend on what you mean about the cleanup, what part of the cleanup process you're referring to. The dead cell removal beads, they're conjugated to NX and V, so it recognizes phosphatidylserine on the surface of the cells. That's normally on the inside of the cell membrane. In cells which are apoptotic or along the further along the death pathway, even from necrosis, it does get flipped to the outside. There are other situations where it can be flipped to the outside, and in that case, they can be recognized. So the dead cell removal kit itself should only recognize cells with phosphatidylserine on the outside. So it should not affect the cell growth. It should just be getting rid of cells which are already damaged. But other steps along, for example, RBC lysis is something that actually we are concerned about in a sense. So it's any of the RBC lysis solutions, including our own, use osmotic pressure to burst red blood cells. They're also going to stress any other cells which are in there. And it can cause increased expression of phosphatidylserine on the outside of white blood cells. So when used in combination with the dead cell removal kit, you may see loss of some white blood cells just because they are stressed by the lysis solution expressing phosphatidylserine on the outside of the surface, and then depleted by the dead cell removal beads. So because the cells are stressed by RBC lysis, or can be stressed by RBC lysis, that may affect their growth. So we are hoping to actually test perhaps alternatives to RBC lysis for this. We have magnetite beads which will specifically target and can be used to remove red blood cells from samples. So that's something we'd like to look at in terms of how that might be used for single cell prep. Yes, there may be some effects, but it's going to depend on the specific treatments.
All right, next question. Have you seen changes in cell type composition through various cleanup steps? Some cell types may be more fragile or likely to clump together.
Yeah, absolutely. And building on what Murray said, in this particular study that we did, the biggest change we saw was that the white blood cells might have been impacted by the red blood cell lysis in subsequent cleanup steps. So we saw a reduction in the fraction of total cells that were immune in origin as we proceeded through the sample prep workflow. I imagine, and that was a major type we kept an eye on in this particular experiment. Again, this was a very simple and controlled tumor model, so there weren't that many different types of cells present. It would be really interesting to look at this on primary tumor samples with more stromal involvement and also see what the impact of the additional cleanups would be. But I'm not surprised. You know, handling cells, as we all know, every time you spin and wash a tube of cells, you're going to lose some cells, and I wouldn't be surprised if there is bias there. Some suggestions, if you know what cell types you're most interested in, and you know conditions that make them the most happy, you can keep your cells in a different media. You don't have to use PBS with BSA. You can keep your cells in culture media with serum. You could spin them really gently in a swinging bucket centrifuge. So there are ways that you can tweak the workflow to preserve the cell types you're most interested in.
Okay, next question. Does tissue storage solution from Miltenyi stabilize nucleic acids just like RNA later?
I'll take that one as well. So the solution, as we've tested it, what we've done is stored tissues in it for transport, and then dissociated them at the endpoint, and looked at cell viability and cell recovery. And on those measures, the results are excellent. So we haven't specifically looked at RNA, although we're getting a lot of very healthy cells, so we expect the RNA to also be good. And I would point out that the tumors used in this experiment that Jill was just referring to, they were shipped to us, so we didn't have in-house animal facilities. The tumors were isolated by Charles River and shipped to us on the tissue storage solution. And certainly we had no issues that I'm aware of in terms of processing the tissues, or the RNA for the single cell sequencing.
Yeah, that's right. They were shipped across the country overnight. So we didn't have the comparator point, which was freshly after excising out of the mouse. But again, we got really nice data sets from the samples that were shipped in the tissue storage solution.
All right, thank you. Next question. What were the viabilities in different tumor types after filtering only and after the extra cleanup step?
I can take that question. I, off the top of my head, do not remember exact numbers for each of the 3 tumor types, but I can give you a range. Post just dissociation, the viability ranged from somewhere 40% to 70%, And like I mentioned, and like Jill mentioned actually in the talk, that there was one particular melanoma tumor that we received that had very low viability, which is why we saw 40% viability over there. So the range post-dissociation was somewhere between 40 to 70, and we cleaned it up with those different steps that she mentioned and took it up to above 75%. Most of them went up to 80%. The tumor that had 40% viability, we got it up to 75%. So that's the range of viability that we saw post-dissociation and post-cleanup.
Very great, thanks. Next question. What temperature should be used while dissociating tissues?
I can take this one. This is Cole. This one would be dependent on the tissue type. We have an array. Miltenyi has an array of tissue dissociation kits specific to things such as lung, umbilical cord, lamina propria, tumors, adult brain, neural tissue. So depending on the tissue type, there is both a designated enzyme cocktail as well as a step-by-step protocol, which is designed for the GentleMax. So that will specify both the enzyme mix, the protocol type, and then all enzyme kits are optimized to perform at 37 degrees, which the heating elements on the GentleMax instrument will apply consistently over the course of dissociation for that particular tumor or tissue type.
All right, thank you. Next question. What modifications are needed for neurons for brain tissue?
I can address this one. I know 10x has done a fair amount here. When it comes for dissociating to get neurons, this is going to be, if you want to get single cells, single neurons, then you could use that. We have our neural tissue dissociation kits or our adult brain dissociation kits. And, these I'd recommend seeing what's going to work best for your animal type and the age, whether they're below or over P7 in age. But so that's going to, just following the Miltenyi dissociation protocol will get you to a single cell suspension where you can then isolate. We have an isolation strategy, kind of a depletion or a negative selection strategy for isolating neurons. So that would be the means to go about getting single neuronal population. Otherwise, I know 10x has done a considerable amount of work to also validate a process for single nuclei isolation from neural tissue to do sequencing of neurons. And I don't know if Jill or Sharmila, if you guys want to elaborate on that.
So yes, thank you, Cole. So we in our 10x DPs, we have a protocol for isolating neural cells from embryonic brain, which is embryonic P18. We have also nuclei isolation protocols that's there for embryonic brain as well as adult brain, and we have seen that the nuclei data corresponds very well with this, matching similar tissue data, so cells from the brain. We, as of now, as per our protocols, have not tested internally isolation of cells from the adult brain. But then again, as Cole mentioned, they do have a kit where they do dissociation of the adult brain.
Yeah, I'll add a little bit onto that. A lot of people are studying neural samples. A lot of our customers and a lot of people in general are doing single cell RNA analysis on different neural populations. And I think there are two major trends I see, at least in the literature thus far. One is to just go the route of isolating nuclei, which gives you a nice representation of different cell types within the brain, and sometimes you can sort with a new N labeled antibody to get just the neuronal nuclei. And if you look at, I think, mousebrain.org, there's a publication from Sten Linderson's group that just did a comprehensive atlas of different cell types in the mouse brain. When people go the whole cell route, people often follow that with an enrichment for one specific cell type. I think because the cell types in the brain are so diverse and varied, it's really hard to get an unbiased, dissociated adult brain sample where you still have every cell type represented. Usually the sample prep protocols are tuned towards the type of cell that you want to isolate. So Miltenyi has some very specific products and specific workflows that they have made available. Mousebrain.org, I believe. I wish I looked up that website before getting on this call this morning. But if you Google Sten-Linderson mouse brain, you can probably get to that website, and you can see the different protocols that they use for each of the different regions of the brains that they looked at.
Yeah, and if I could just add one more thing, is building on that, for neurons especially, the more mature neurons with really long axons, very delicate. So any sort of dissociation protocol is going to, for neuron isolation, is going to bias the sample towards the more immature neurons, because just they have such long processes that are very delicate that any sort of physical dissociation is going to damage a lot of the neurons with longer processes. One other thing I don't think was specifically mentioned, but one big difference with the neural cells to other tissues is myelin, and that's why Cole was referring to P7. We have a couple different options for removing myelin. Beads, which specifically target myelin and is used over a column. That's good for younger mice that don't have below P7 that don't have a lot of myelin. But with older mice, with the adult ones, then it's too much myelin. It'll clog up the column if it's done that way. So in those cases, we use a density centrifugation, a debris removal solution to get rid of that. There are reasons, depending on sample size, that might bias for choosing one over the other. And for that, really, it will depend on the specifics of the experiment. So we really recommend, if you're doing that or have questions about it, to contact the support team here at Miltenyi, and we'll walk you through what factors are important to consider and why you would choose one over the other.
All right, thank you. Next question. Can you share more information about your single-cell immune profiling platform? Can you measure immune cell activation states with that platform?
I think for that one, I'd have to refer to our product website for more information.
All right, thank you. The next question. Our fraction of reads in the cell is usually around 50 to 60% for human tumor dissociation. Are our data still useful or biased?
I think I wish I could talk to the question asker. I think what I would encourage people to do always in this case is look at the cell clustering and look at the loop cell browser results, or look at the results in Seurat, however you want to look at it. You can still get useful clustering and useful data when the fraction of reads isn't optimal. Maybe there could be some artifacts in your data set, but certainly, especially when you get closer to 60%, you're probably getting accurate representations of cell types. I would encourage you to go through, look at markers that you think would be expressed, make a judgment for yourself based on what you know about the biology of the sample, if that's well represented or not. Your samples do not have to be perfect to give you good data.
All right, thank you. Next question. If I use flow sorting for CD45 positive cells, do I need to remove red blood cells or dead cells ahead of sorting?
Nope. It should probably work just fine.
All right, thank you. Next question. Can we freeze the single cell suspensions? If so, should we use the standard chaotropic agent like DMSO?
Yes.
Yes.
Yes. That's the long and short of it. Yeah, you can freeze the cell suspension as you would cryopreserve any type of cell that you'd want to reculture later, and it should work pretty well. Again, super fragile cells. Will any freeze thaw have an impact on cell representation? Yes. But this is still a really great path, and I think the leading vendor of commercially dissociated tumor cells, they use a similar procedure where they slowly freeze the cells with a cryopreservation agent.
All right, next question. Do the enzymes in the tumor dissociation kit affect the expression of surface markers on the cell population when you want to do any FACS validation?
I can take this one. So this is where the Miltenyi R&D team does an extensive evaluation during product development as well as additional validation, where whenever making these enzyme kits, basically they screen across a wide array of cell surface proteins, epitopes, to see what's impacted and what's not. And then ultimately, the enzyme cocktail and concentration are chosen based on how to maximize preservation of those cell surface markers and epitopes. So that's the kind of one of the values of these kits is the work that goes into enable someone to then use flow cytometry, and since we both also do flow cytometry here at Miltenyi as well as cell separation, it is important for us also to preserve those cell surface epitopes.
And just to add, for each of the tumor dissociation kits, we have with the product page there is a list of all of the epitopes we've looked at. Most of them are unaffected or not detectably affected. There are a few that are highlighted as slightly reduced, and some that are ablated, essentially. We've done the best we can, but there are still some to be take consideration of. But there is a complete list available, and so you would factor that into when you're doing your analysis.
And if you have any specific questions about a particular epitope that you're concerned about or wanting to see if it is preserved, you can always reach out to the Miltenyi Science support team, and they can look in to see if that epitope is in fact affected or not.
All right, thank you. Next question. Do you have data on fixed single cells?
Yes, we have data on fixed single cell. At 10x, we have released a DP for fixing single cells using methanol fixation. We have used cells isolated from embryonic brain and fixed them. We've also tried cell lines, and we do observe a certain loss in RNA content, but even with the loss, the data that we get out of fixed cells is good to represent all the different cell types, and the data looks similar to what fresh cells would look like.
Yeah, I think the caveat there is that this method right now is best suited towards larger cells or cells that have a higher RNA content to start with. Methanol fixation is challenging for immune cells and low RNA containing cells. There's recently a publication from one of our customers which has worked on an improved implementation of methanol fixation for PDMCs. So if you're interested in smaller or immune cells, I would encourage you to check out that publication. It's not something that we have validated internally yet, but the data look really nice.
All right, next question. Do you clean out, in quotation marks, some specific cell types with the dead cell removal magnetic beads, which are present in a higher number in the not cleaned up sample prep?
Again, I think, oh, sorry, the only thing we saw was a slight reduction in the immune cell representation.
Yeah, exactly. In theory, it shouldn't. Any cells which express phosphatidylserine on the surface will be depleted, but if you have a population that is more sensitive than to whatever condition you're using, then it will be targeted by the dead cell removal beads.
All right, next question. What is your experience with human tumors, which are more heterogeneous than syngeneic mouse tumor models?
So when it comes to human tumor models, we presented some data on human tumor cells that we've purchased that have been previously dissociated. So we don't have a pipeline for getting fresh human tumor samples in-house. So we presented data showing that you can detect an even more diverse population of cell types. And that's our experience internally, and you can download an application note on our website. But I'll kick this back over to Miltenyi, who they do have expertise and experience in dissociating human tumors.
Yeah, just kind of as a high-level mention of our ability to work with human tumor tissue. We have two kits. There's a mouse tumor dissociation kit and a human tumor dissociation kit. And kind of the rule is that whatever species, whether it's mouse or human, that the tumors are from, then that's the tissue kit you would utilize. And I say that because obviously that makes sense if it's a primary human tumor, but if it's, let's say, a PDX mouse model where the tumors inside of that model are human, in that case, you would also use the human tumor dissociation kit. So that's how you would then get the single cell suspension from those tumors. After that, then it's kind of, left up to what the researcher, what they're going to do with it, whether it's additional cleanup, additional cell separation or flow cytometric analysis or single cell sequencing, and all of that can be done downstream.
Yeah, the one exception to that rule might be if you're looking for infiltrating cells into the tumor that are from the host. So in the case of tumor infiltrating lymphocytes in a human tumor in mouse, in that case you might want to, just for epitope preservation, because the tests that have been done for each kit is for the epitopes on the cells from that species. So to be assured of that, if you're looking for the mouse TILs in a human tumor that's been in a mouse, you probably want to use the mouse tumor dissociation kit just to make sure that the preservation of that is consistent with what we have seen.
Great, thanks. We're coming up on the hour now, so I'm going to try to squeeze in 2 more questions here. The first question is, I'm getting human tumor samples. Is the Miltenyi RBC lysis species specific? Because the countess counts RBCs as dead cells, and we get very low viability, but the RBCs bring it up. Also, I noticed that only the 70 micron filter was shown. Should we also use a 30 micron filter?
As far as I'm aware, we don't consider the RBCs in the viabilities. I'd have to double check that, but if you're getting significantly lower than what we say we get, then definitely contact us. We'll work through with you what we can do to help. For going through a column, yes, I would definitely recommend filtering through a 30 micron before going through the column. The reason being, the pore sizes in the column are about 30 microns. So if you don't do that, you're essentially using the column as a filter, and it will clog up with anything that's bigger than 30 but smaller than 70 and affect the flow of the cells through. And that could be one, if people are losing cells, that could be one reason.
And one kind of best practice we use at Miltenyi here is after dissociation, we do the initial filtration step that Jill mentioned, where we use our 70 micron smart strainers. But then there's a final filtration process that happens just before the sample runs through the column, and we have these pre-separation filters which fit nicely specifically on top of the columns. And then that's that one final, and you can get I think it's a 30 or 20. Yeah, you can get a 30 micron filter on that one. So that's that final best practice filtration just before the sample goes into column, ensuring that you don't clog the column.
To add on to that, even at 10x internally, we do recommend filtering your samples with a 30 micron before loading it onto the 10x platform. This just ensures that you have a single cell suspension and you're avoiding clumps or cells aggregated together to get better sample quality.
All right, thanks. And one last question. I worry about the time required for processing. Do you have any data that explores changes in individual gene expression over time in the course of processing? Are there viable options for fixation at the tissue or the dissociation steps to allow batching of single cell capture runs on the chromium?
So I think you're right to be concerned about the processing time. There's sort of, tissue dissociation. There will be impacts, biological impacts of these procedures. That the advice that I would give is, use the procedure where you're processing with a minimum amount of time possible. I like the Miltenyi dissociator tube because it physically helps with dissociation. It sort of opens the tissues up more so that the enzymes can act quickly. So minimizing time is a really good idea there. When it comes to specific cell types or specific tissue types, I would pay attention to the work that the Human Cell Atlas community is doing. There is a whole community of investigators. Each one is focusing on a different tissue and really optimizing the dissociation for that particular tissue. So it's really early days right now, but I would follow along with that over the course of the year. I think you can even see some of their work on protocols.io, but there will be, if you want really, really optimal results from each tissue type, you can see if a colleague in your space is working with the same tissue type would be my recommendation.
To add on to that, this also depends very much on the experiment you are targeting as well as the tissue type. If you are looking at some sort of signaling that is a very short span signaling, this might be affected. Obviously, it totally depends on specific genes that you are interested in looking at and how quickly they change their expression pattern based on the different conditions. But to add to your question, yes, we do have fixation protocol as we mentioned before, and as of now, like it is for cells with higher RNA content. So if you have the capability to fix cells, definitely avail that route. If you're worried about change in transcriptome in your different samples due to processing.
And my advice when it comes to batching right now is to process the samples as soon as they're ready. Don't wait until you have a set of 8 to run the chip. Run the chip as soon as one sample is ready. Then you can batch them after the gems are generated, and you can do the library prep in parallel afterwards. So that's a good approach, so you can still maintain that you maintain the minimal workflow time on the sample prep. Then once you get the cells encapsulated, there are many more stopping points. And I know that's a frequent way in which our customers handle the batching question.
All right, thank you. That brings us to the end of our session for today. I'd like to thank our 10x-perts, Jill and Sharmila, and our Miltenyi Biotec special guest, Colin Murray, and especially to our attendees for some great questions. We'll be posting a recording of this session on the 10x community shortly. If we didn't get to your question, or if you have a follow-up question, we urge you to post it on the 10x community in our single cell forum at community.10xgenomics.com. And finally, we have a couple more user group meetings scheduled this year in Toronto, Paris, and London, and we invite you to join us for a great day of talks, networking, and 10x-pert discussions. You can find out more on www.10xgenomics.com/UGM_2018. Thank you, and have a great rest of your day.